introduction, and thank you to everyone who is logged on. I'm so excited to get to share with you what I've been up to for the past 5 years. So as I'm sure you're all aware, the world is full of a pretty remarkable array of creatures, and some of the most outlandish traits and behaviors that we get to appreciate are the product of sexual selection, because in some way these characteristics enhance the mating success of the individuals that carry them. But of course, organisms are also subject to natural selection, and we know that specific sexually selected traits can be constrained by biotic or abiotic selection. For example, a peacock's long tail may hamper its ability to fly. A brightly colored lizard might be more visible to potential predators. But what's less well studied and understood is how more general environmental conditions can influence the strength of sexual selection outside of the reproductive context. So in other words, how is sexual selection influenced by natural selection that's occurring on traits and behaviors that are not themselves the products of sexual selection? Natural selection can occur through the abiotic environment, and temperature is an abiotic selective force that we've thought about a fair bit. And just by looking at organisms that are adapted to extreme cold or heat, we can get a sense for the clear physiological consequences that freezing temperatures or heat stress can have on organisms. Another source of abiotic selection is precipitation, and we're used to thinking about lack of precipitation and adaptations that organisms might have to go for periods of time without access to water. We also know that drought events can be really powerful evolutionary drivers shaping organisms, but what's a bit less well researched is whether and how too much rain can influence the fitness of organisms. So rain as an abiotic selective force might be especially relevant in the tropics. This is an example of what is common throughout much of the tropics, where temperature is very constant throughout the year, and instead seasons are defined more by the precipitation amount. So there's periods of year when rainfall is quite heavy, periods or dry seasons when there's very little rainfall. Rain is also a very fundamental characteristic in defining the plant communities that we find throughout the tropics. So increasingly, in recent years, there's been a growing handful of studies that have recognized the important role that rain can play in shaping demography and life history of tropical endotherms. Organisms that are wet and dealing with slightly colder temperatures may have more difficulty thermoregulating. Rain might contribute to availability or accessibility of food resources, and the combination of these and other factors may result in influencing the ability of organisms to survive and produce offspring. So this is sort of the big overarching question that my PhD is centered around, is how natural selection through precipitation can influence sexual selection, specifically in tropical birds. And so I tackle this question at multiple biological levels, starting out at the clade level, looking at patterns of morphology and relationship with abiotic environment, then narrowing down to looking at multiple species within a specific location and how interannual variation in rainfall can influence their demography. Then within a species, but looking at different populations of that species, trying to understand how different climatic conditions can constrain reproductive skew and sexual selection in those populations. And then finally focusing in on a single population, testing hypotheses about drivers for individual variation in behavior at display sites. So starting off at the clade level, what better group to study how abiotic selection might influence sexual selection in the tropics than with neotropical manakins, which are these lovely small fruit-eating birds that are found throughout Central and South America in a range of different types of tropical forests. What they're probably best known for is their mating system. So males gain all of their reproductive success through the displays that they do for females at these designated sites known as leks. So females visit the lek, assess multiple males. Usually they settle on just a few out of that population to mate with. And then the females, then once they've chosen their particular mate, will go on to do the hard work of building the nest, incubating the eggs, and raising the chicks. So this is a system in which there's a lot of potential for sexual selection. And we see evidence of that in the dramatic array of displays that males of different species do. And you'll notice that males in all of these species are very brightly colored. Interestingly, females, without exception, are all a lovely cryptic green. But there's another way in which males and females of the same species can differ, and that's size. So size is a very fundamental characteristic of organisms and dictates everything from how much energy they require, how fast metabolic reactions can occur, what sorts of foraging strategies are appropriate, what types of habitat they can live in. And so given these constraints, it's sometimes surprising to realize how different male and females can be in size. In a sexually selected context, we're accustomed to thinking of males that are bigger and larger, potentially being able to defend access to resources, or females. But there's also potential for advantages to larger females, being able to produce more offspring. Males and females may partition out the resources that they're searching for. They might have different thermoregulatory requirements. But usually the direction of size dimorphism within a group is conserved. So in amphibians, females are generally larger. In mammals, males are generally the larger sex. And in spiders, for example, females are often bigger than males. But in manakins, the direction of sexual size dimorphism varies across the clade. So there are species in which males are larger than females, and there are species in which males are smaller and lighter than females. So this is really the question of my first chapter of trying to understand why sexual size dimorphism varies across the manakin clade and whether that's due to differences in sexual selection or differences in natural selection exerted by abiotic conditions. So if sexual selection is driving sexual size dimorphism, we can look to these displays where some species have aerial display movements, other species are doing more perched postures. And if sexual selection is driving sexual size dimorphism, if we look at our range of species from species in which males are smaller than females through to where males are larger than females, we might predict that agility and sexual selection through female choice might select for males to be smaller in species that are doing more aerial display behavior. So if we think of a male manakin as basically a gymnast, being small and compact might allow him to perform these movements more efficiently and effectively, as opposed to species in which males are doing more perched behaviors and fewer aerial behaviors, they may not be selection for them to be small. The abiotic conditions that manakins are contending with may have greater consequences for males because they spend so much time displaying for females. And so if the environment, such as thermoregulatory challenges, could be influencing size, it's quite difficult to maintain a constant body temperature, especially for these really small birds. It can get quite hot at low elevations. Even in the tropics at high elevations, it can be pretty chilly. And then on top of that, rain makes thermoregulation more difficult. So if natural selection is driving sexual size dimorphism via thermoregulation, then we might expect that at these more extreme high elevation cold environments and low elevation hot environments, the energetic cost of staying warm may select for males to be the same size or larger than females in those places. Another way that the abiotic environment can influence size is through resource constraints. So manakins eat fruit, which means that they need to eat pretty constantly to maintain their energy levels. And we know that really heavy rains can actually restrict manakins' ability to fly to access those food resources. And so if resource constraints are shaping sexual size dimorphism through the abiotic environment, we predict that in species that are living in less rainy environments, males can be small because they can easily access food whenever they want to, whereas in species that are in slightly rainier environments, perhaps there's selection for males to be a bit bigger and bulkier to be able to withstand periods of fasting during storms. In addition to mass, there are other morphological traits which could vary between the sexes. So wing length, longer pointed wings are a bit more efficient for long distance flight, but having rounder, shorter wings can confer a bit more maneuverability, which of course may be good for males who are doing these displays. Tarsus or leg length is also a really good, consistent measure of structural size and might also be under selection in species that are doing bowing behaviors or leaping in their displays for females. So as Alice mentioned, throughout my PhD, I've been fortunate to be part of an NSF-funded research coordination network that's focused on understanding sexual selection in manakins. And so through some wonderful collaborations with other manikinologists, I was able to compile morphological data for 22 different species of manakin, mass for all of those species, and then wing and tarsus measures for a subset of those. I accounted for differences in evolutionary relatedness amongst these species using a new manakin phylogeny. And then I measured sexual size dimorphism following Lovich and Gibbons. So when males and females are the same size, the index is 0. It's a negative value when males are smaller than females, and a positive value when males are larger. For the environmental variables, I used localities that had been collected from specimen records for a 2009 paper. And so for the coordinates of each of those, I extracted 13 different precipitation, temperature, and elevation variables. And then I collapsed those down into 2 principal components. The first one basically consisting of temperature and elevation, and the second being variables related to precipitation. For agility, I modified a method that was used in a 2015 paper and basically just tallied up counts of the aerial display behaviors that were included in a species display repertoire. So any behaviors that were part of display that were perched didn't require the male to defy gravity in some way weren't included in that score. I then did a phylogenetic generalized least squares regression for each of these 3 different axes of size separately. And if we look first at variation across these 3 different traits in these different indices, you see that there's quite a bit more variation in the mass than either the wing or the tarsus right off the bat. So getting at this question of what drives sexual size dimorphism in mass, we found overwhelming support for agility score as the best predictor of patterns of sexual size dimorphism across these species, even more so than additive or interactive effects with environmental variables. So if we look at that relationship here on the y-axis is our index of size dimorphism, so males larger, males smaller, males and females the same size at this dashed line. And then on the x-axis is our agility score, so down here will be species that have few aerial display elements, and then over here species that have more. That relationship between these 2 variables shows that males do tend to be smaller and lighter in species that are doing more aerial display behaviors than in species that are doing fewer. So this seems to answer the question that what is driving manakin sexual size dimorphism in mass is sexual selection for agility, such that in species with more aerial display elements, males are smaller than females, and in species with fewer aerial display elements, males tend to be the same size or larger than females. For the other axes of size dimorphism that we looked at, we didn't find any effect of the environment or agility on tarsus length, but we did see that in wing length, males tended to have longer wings in species that were found in more rainy environments. And so this, when we look at the number of species that are either larger or smaller than female for the subset of species for which we have all 3 measures, what's interesting is you really see that there are multiple species in which males are larger than females in wing and tarsus, but lighter than females in mass. So it seems like there's really a role for both sexual selection and abiotic conditions in shaping the morphology of manakins. Sexual selection given its strong association between agility and mass, but abiotic conditions, specifically rain, are having a role in shaping wing length of manakins. So these role of sexual selection and abiotic selection at this higher taxonomic level is very exciting. But it's also important to realize that sometimes these patterns and associations that we see with abiotic conditions that we see at higher taxonomic levels are likely driven by survival and demography that's happening on a more local scale within populations and communities. And until recently, we haven't really had a clear framework for understanding this relationship between precipitation and fitness. So I'm not going to get into a ton of detail, but in the past year or so, we've been working on articulating this idea of the hygric niche. So species occupy a range of different precipitation conditions. Some conditions will be optimal and they'll have maximal individual performance. At other locations that are wetter or drier, they may still be viable, and of course we won't find those species outside of their hygric niche. So if this is the case, we would predict that in a community that's an assortment of multiple different species at a specific location, the response of those species demography to increases or decreases in rainfall would depend upon where that site is in that species' respective hygric niche. So this is essentially what I did in my second chapter, is tested this idea by looking at demographic responses of different species to increases and decreases in rainfall from year to year. I tested this idea at a beautiful part of Costa Rica known as Rar Avis, which is a really lovely mid-elevation primary forest. And we predicted that the estimates of apparent survival would vary year to year based on rainfall, but the direction of those variation would likely depend upon the species and where it was at in its respective hygric niche. So across the 7 years that this data was collected, annual rainfall at this site varied between 7,000 millimeters and 9,000 millimeters per year. And Alice and Dr. Megan Jones were able to go out and capture birds in a standardized manner during those years, so that I was able to construct Cormac Jolly Seaver models to estimate annual apparent survival for 7 species. 4 of these species had not previously had published demographic estimates, so exciting to get some initial info about them. And so once we had those models and estimates of apparent survival, we wanted to predict what the relationship between estimated apparent survival and rainfall would be. In 2 species, apparent survival was not affected by precipitation. There was no relationship. They didn't have higher or lower estimates of apparent survival based on how much rain fell in that year. In 2 species, estimates of apparent survival actually increased, so they had higher probability of survival in years that received more rain. And for 3 species, their probability of apparent survival declined after years with more rain. So they had a negative relationship with increased rain. So as we had predicted, rain was important in shaping demography, but the directionality and implications of that effect varied by species. And a lot of times understanding the mechanistic links between survival and precipitation are difficult without having a bit more context about the behavior and life history of those species to understand what's being affected by these different rainfall conditions. So knowing that that the relationship between demography and rainfall does exist, for the remainder of the talk, I'm going to be focusing on 1 species, the white ruff manakin, to try to get some insight into some more of the nuances into how exactly abiotic constraints might influence sexual selection in this species. So Corapipo altera, white ruffed manakins, are delightful little birds. They're between 10 and 13 grams. As most manakins, they live for quite a long time, can be more than 10 years. They're found throughout Central and Northern South America, and they like to hang out in mid-elevation, often pretty wet tropical forests, primary tropical forests. In this species, males do their displays on fallen mossy logs, and so each fallen log display site has its own owner or dominant male. Sometimes we call him the alpha. But these display logs frequently are sort of clustered within auditory range of other display sites, which would all have their own dominant alpha male. And so this collection of display sites that are all sort of within earshot of each other are what we call the lek. And display sites can have a single male owner. In some cases, display sites will have multiple males associated with them. But in those cases, there's always a clear dominance hierarchy, and there's always a dominant male who's the one who gets to mate if a female is interested. In this species, males reach their adult plumage after 4 years, but it can still be several years before they attain dominance at a display site. And there's still no guarantee that females will come visit their logs and be interested in mating with them. So it's quite a protracted life history with a lot of potential for reproductive skew. Although this species was not one that had changes in estimates of apparent survival from year to year based on rain, we do still know that in this species, rain events and storms are physiological stressors, and manakins show evidence of fasting and mortality after big storm events. Males also often migrate to lower, less rainy elevations during the non-breeding season to escape the heaviest rainstorms. However, they do bear consequences of that choice, as when they return for the next breeding season, they tend to lose status, and females actually prefer males that did not migrate. Although we didn't see any year-to-year variation in estimates of apparent survival, the overall average probability of survival for male manakins at this site is quite low, 0.62, especially as compared to estimates of apparent survival that have been calculated in other populations. And when we look at the precipitation regime between those two different sites, it's pretty strikingly different. So in this population where males have a probability of apparent survival of 0.9, they're dealing with less than half as much rain on a yearly basis than the birds up at Rara Avis. So my third chapter is really focusing on testing the idea that these population level differences in demography that may be driven by environment could influence reproductive skew and sexual selection of these different populations. We predict that differences in mortality can strongly affect variance in reproductive success, that is, the proportion of males that have an opportunity to mate. So if we imagine two populations of white ruff manakins, in a population that's not dealing with much rain, has very high survival, then we predict that the males that are most attractive and monopolize most of the mating opportunities have an opportunity to survive year after year and continue to overwhelm the gene pool. And so in that population, there is higher reproductive skew. So there's a lower proportion of males at that site that have an opportunity to successfully reproduce. Conversely, in rainier environments where there's higher mortality, we predict that there's going to be more turnover in males that are dominant and favored. So even though one male may be the most attractive and monopolize most of the matings in one year, the following year he may have had to migrate, he may have died. There's an opportunity for another male to have some reproductive success. And so in that population, we predict that there's going to be reduced reproductive skew, and a larger proportion of males in that population are going to have an ability to reproduce. So to test this, ideally we would go out and take genetic material from a bunch of cute baby manakins, but that's challenging under the best of conditions. And these guys like to nest 8 meters up in the canopy, so it's a bit more challenging. So instead, we took a somewhat novel approach of using the genetic diversity of sex chromosomes and autosomes to measure reproductive skew indirectly. So in birds, males have 2 Z sex chromosomes and females have a Z and a W. And of course, both sexes have the full complement of autosomes. So there's basically 3 Z chromosomes for every 4 autosomes in a population. So if there's no reproductive skew and all males are having an equal opportunity to reproduce, the genetic diversity of the Z chromosome relative to autosomes should be about 0.75. However, if some males don't get to reproduce, then the genetic diversity of the Z chromosome is going to decline relative to the autosome. So what does that look like? If instead of cute little white ruff manakins, we think of them as more sets of Z chromosomes, then in our population with higher skew, there's going to be reduced genetic diversity of the Z chromosome, a lower effective population size of Z chromosomes in that population, as compared to a population where there's more turnover, a higher proportion of males are producing, and there's a greater effective population size of Z chromosomes relative to autosomes in that location. So this is the question that I tackled for my third chapter, asking how reproductive skew as a measure of sexual selection is influenced by environmental variation. So this metric of Z relative to A diversity has been used in birds to look at differences in reproductive skew between species, but this is one of the first instances of looking within a species whether there's differences amongst populations that are contending with different selective regimes. But thanks to the expertise of the manakin genomics group, in particular Chris Balakrishnan, We were able to sequence and assemble a reference genome for the species, and then use a cactus alignment to identify portions of the genome that were on the Z chromosome and portions of the genome that were autosomal. So to get this genetic material from sites that were experiencing a range of different environmental conditions, in Costa Rica, there's a central mountain range that sort of divides the country into a Caribbean slope and a Pacific slope. And on both sides, there's a biogeographic gradient from more dry conditions in the northwest to slightly more wet in the southeast. And so I sampled along these two slopes. And I've colored the Caribbean slope sites in shades of blue and the Pacific slope sites in shades of red, just to try to keep track of that. Traveling and getting to all of these places was a great adventure, a lot of fun, and a really good time. And ultimately, we were able to get some data, so it was all good. The sample sizes at these different sites varied a bit, just depending on how long we could stay and what sort of success we had. But ultimately, from the blood samples that we collected from adults, I was able to extract DNA for 158 individuals and send it off to the University of Minnesota for a rad sequencing approach. The environmental variables that we wanted to look at that could be influential in demography and therefore influence reproductive skew included temperature because of potential thermoregulatory costs, which could influence survival. And then, of course, we wanted to look at mean annual precipitation for these sites, knowing that there's quite strong differences in demography at sites that have different total annual precipitation. And we were also interested in how the distribution of rainfall throughout the year could influence reproductive skew. So white ruff manakins breed during the dry season, but on the Caribbean slope of Costa Rica, there's still a fair bit of rain that falls during the breeding season. So we were interested in whether that might increase the cost of display for males and therefore influence reproductive skew. So we measured Z relative to A just in males and took the ratio of Z nucleotide diversity and autosomal nucleotide diversity measured across 10 kilobase pair windows. Because there was a variation in sample sizes at all of these sites, we wanted to have some measure of error. So what we ended up doing is we measured Z relative to A at the maximum N sample size for each site, and then we rarefied down measuring N for each subsampling and measuring Z relative to A for each sample size down to N equal to 10. And there's one site that we only were able to capture 6 males, so it's just a point estimate. And so then just to remind you and orient you, so these sites with higher values of Z relative to A nucleotide diversity are indicative of reduced skew, while lower values are indicative of increased reproductive skew and potentially sexual selection. So the relationship between Z and A and temperature wasn't really there. Also, surprisingly, there wasn't any clear association between annual precipitation and Z relative to A nucleotide diversity. However, there was a relationship between Z and A nucleotide diversity at sites. It was highest at sites where rainfall was the least seasonal. So in places where rain is falling throughout the year, this may increase the cost of display, reducing reproductive skew and elevating that Z nucleotide diversity relative to autosomal nucleotide diversity. So it seems like there is the potential for seasonality of rainfall to indeed constrain the strength of sexual selection and reduce reproductive skew across populations of manakins. But the environmental differences are somewhat confounded by the slope level differences. So there's quite strong differences between the Caribbean and Pacific slope in seasonality, as well as in these measures of Z relative to A. So previously, Corapipa have been described as subspecies on the Caribbean and the Pacific side based on morphology, but there haven't been any genetic characterization of that yet. And so I wanted to see if there were any underlying genetic differences between these two groups that could contribute to the slope level differences that we see in Z nucleotide diversity relative to autosomal diversity. So first I did an admixture analysis to look at differentiation. So each column here is basically an individual, and the color of it corresponds to its probability of assignment to a specific cluster. And so as you can see on the Caribbean side, And on the Pacific side, there's evidence that there's recent and potentially ongoing gene flow happening between these sites. But the distinction between the Caribbean side and the Pacific side is quite stark. So there's no evidence that gene flow has happened across slope for a while, probably. Looking more quantitatively at differences in allele frequencies, as we'd expect, there's quite low distinction between sites that are adjacent to each other, suggesting there's still gene flow happening, and there's more distinction, more differentiation between sites that are on different slopes. Within populations themselves, there's some slight differences in measures of genetic diversity. So there's a bit less inbreeding apparently in sites on the Caribbean slope as compared to sites on the Pacific slope, though errors do overlap quite a bit. And there also seems to be a bit more heterozygosity of individuals that are at Caribbean slope sites. But if we look at mean nucleotide diversity across the whole genome, there's no emergent pattern based on slope. So it seems like currently effective population sizes of these two groups are fairly similar. So definitely disentangling the effects of environment and demography from other evolutionary processes that are ongoing or historic is challenging. And so I'm hoping to do a bit more demographic modeling to try to see whether some historic population biogeography might help us determine these difference in metrics. And of course, being able to sample at some additional sites would also help strengthen our inference. But overall, finding this relationship between Z relative to A in a direction that we would predict with clear underlying differences in demography is really exciting, as it has implications for the strength of sexual selection varying in these populations that are dealing with different abiotic conditions. And I think it's also a very exciting potential metric that could be used in other systems to try to understand where there might be variation, reproductive skew, and relationship with other constraints. So for my final component, I wanted to focus within a population and understand, test some hypotheses about what might be driving variability in individual behavior at display sites. So this is focusing on the beautiful Parque Nacional Volcán Tenorio in Costa Rica. And even though males at this site are obviously experiencing identical environmental conditions, we still see dramatic variability in what they're doing day to day at their display sites. So some males are super attentive and successful. They're constantly at their log. Females visit frequently. Some males are quite attentive and they are around quite a lot, but they don't get nearly as many female visits. And then there are some males that just put in the bare minimum. We rarely see them at display logs. They get few, if any, female visits. So I wanted to understand why some males were rock stars and others were slackers. And so one hypothesis is that it's just an inherent intrinsic characteristic of the individual. They vary in their quality and condition, and therefore how much effort they can put in at their display site. We also considered whether it might be due to competition. So perhaps some males need to display more and put in more effort to maintain ownership at their particular display site. And another thing that we considered is whether there might be cooperative adjustment of behavior of males based on what their neighbors within the lek were doing. So to test these three hypotheses, I did two experiments in which I altered the perceived social environment. In the first, I set up speakers and simulated an increase in mannequin display around a focal log. In the second, I temporarily reduced display activity within the lek by covering neighboring logs with big palm fronds and preventing those males from displaying on their logs. We predicted that if it's male quality or condition that's driving the variability in behavior that we see, that we wouldn't see any change in response to these manipulations. The males would just carry on doing what they were going to do anyway. However, if competition is driving variability in behavior, then we predicted that in response to this perceived increase or potential increase in competition at around the focal log, males would increase their activity to maintain ownership. And if cooperation is what's driving variability in behavior, we predicted that males might reduce the effort and activity that they're putting in when we have speakers turned on, increasing the signal around the focal log. And males might increase their effort or activity if we reduce the display effort that's happening within the LEC as a whole by covering up nearby logs temporarily. So I did this experiment over 2 years. In the first year at 10 different display logs, we set up 3 speakers, and over the course of 8 days, subjected the male at the focal log to 3 different treatments. The first was just control with no manipulation. Then we played sounds of a non-manakin species from the speakers. And then during the last phase, we were playing the sounds of male manakins displaying and vocalizing. During each of those days, we did both in-person observations and we set up a GoPro to do video observations, and we alternated which of those types of observation happened in the morning and which happened in the afternoon. In the second year, at 7 display logs within 3 different LECs, I temporarily covered up all the logs within the LEC of the focal log. And again, this was an 8-day trial, but with only 2 treatments. So the first was control with no manipulation, and then 4 days of manipulation when the logs were covered. And then again, we did in-person and video observations, alternating which happened in the beginning and which happened later in the day. The behaviors that we measured during these observations included how much time males spent at the log, how many individuals were observed at the log, and how many displays for females happened at the log. As far as what the males themselves did, we grouped those into vocalizations, which aren't necessarily associated with display, but males, if they're near a log, will do advertisement trills. If there's multiple males hanging around, they'll sort of get excited and do their excited trills. And then we have silent display behaviors, which males do very close around in the understory in the vicinity of the log. And then we also had acoustic display behaviors, which males do both away from and at the log, which include mechanical and vocal components. And I'm going to try to play it. I don't know if it'll work, but maybe, fingers crossed. Okay, well, maybe that worked. So once we had collected all of that data, I constructed generalized linear mixed models to test for the effect of treatment on each of those behavioral responses. And I did some initial preliminary analyses to see whether there were other factors that could influence behavioral response. So whether there was differences in our detection when we were doing video observations versus in person, or whether there was seasonal variation in display behavior, or whether there was daily variations in males doing certain behaviors in the morning or the afternoon. So if there was any correlation with one of those factors and a behavioral response, I included it as a fixed effect in the model. I then included a random effect of log identity to try to account for any individual variations. So we're just seeing the effect of the treatment on behavior. For the removal experiment, I also included a random effect of LEC identity and the order in which the log was manipulated. And then finally, of course, a fixed effect of treatment, which for the playback experiment was control heterospecific for the manakin treatment, and for the removal was either control or manipulated. So for the playback experiment, I'm showing in these graphs, the points are the model predictions for that particular treatment, and then the gray points are all raw data values that go into the models. For the playback, we didn't see any change in attentiveness, number of individuals, or displays for females during any of the treatments. So this is consistent with the quality or condition hypothesis. We did see that males actually reduced the amount of vocalization that they projected into the environment in response to increased acoustic signal near the focal log. So this is consistent with the cooperation hypothesis. For the removal experiment, there was no change in the attentiveness or number of individuals at the display log, but there was a slight increase in the number of displays for females. And so these are consistent with the quality and condition hypothesis and the cooperation hypothesis, respectively. And for the other display behaviors during the removal experiment, there was some very marginal increase in number of silent displays in response to treatment, but other behaviors were unchanged, which are overall consistent with the quality or condition hypothesis. So we didn't find any support For the competition hypothesis, males never increased their attentiveness in either experiment. There was no increase or decrease in the number of individuals that were visiting display sites. And the consistency in display rates in males for most of their behaviors throughout the experiment is consistent with the quality or condition underlying some of the individual variation that we see amongst males. The adjustment of male vocalizations in response to increase in acoustic signaling suggests that males may vocalize more as a lek site advertisement than as a display site defense. So males may adjust how much they're signaling in response to what their neighbors are doing. And the increase in displays for females that we saw could be explained by the fact that the males at the covered logs were still able to vocalize on the lek. And so perhaps if with that signal of advertisement preserved, when females visited the lek, they may have just been funneled to that focal log, increasing the rate of displays for females there. Support for both the cooperation and the quality or condition hypothesis may be facilitated by the multimodal signaling that's going on. So potentially females are identifying the presence of leks at a distance using the vocalizations, and then once they arrive on the lek, are assessing the quality and condition of individuals based on the behaviors that they're performing at the log. Complex cooperative associations between males that are sharing a display site is a hallmark of several manakin species. And in these species, males are carefully coordinating their behaviors and are adhering to very rigid social hierarchies. And so perhaps this more loose cooperative signaling at the lek level could help us understand the evolution of some of these more intricate, complex social interactions in other manakin males. So looking back across all of these levels of biology and how natural selection through precipitation might be influencing sexual selection in tropical birds, it seems like there's a possibility that cooperation amongst males at the lek could help them conserve their effort. We see that there's at least putative evidence that seasonality of rainfall is constraining sexual selection across populations of white-ruffed manakins. Within a community, we saw that there was indeed differences in demographic response to increases and decreases in rainfall depending on species. And then at the clade level, we see evidence that both sexual selection and abiotic selection based on precipitation regime can influence distinct axes of size dimorphism in manakins. So I've shown that abiotic conditions in the tropics can drive evolution in previously unappreciated ways through comparative studies and population genetic research. We see that rain that shapes life history and demography can have implications for the scope of sexual selection and evolutionary trajectories of these organisms. I've also provided crucial support for ideas about how rain shapes tropical endotherm fitness. And this is increasingly important as we projected shifts in precipitation regime will likely lead to differences in the selective pressures that these organisms are facing. And finally, as a whole, my dissertation focuses on how abiotic selection can constrain and interact with sexual selection to, through demography and life history, establish the scope of sexual selection outside of the context of reproduction and display. And with that, I have so many people that I would like to thank. My committee for all of their guidance and wisdom, the people at the Division of Biology who make everything run smoothly, current and previous members of the Boyle Lab for all their encouragement and feedback. Of course, the KSU biology grad students who have made living in Manhattan so much fun, are always down to commiserate or celebrate depending on the day. And when I haven't been in Manhattan, I've been so incredibly privileged to live and work in Costa Rica with some really wonderful people who have made me feel at home. And I'm so thankful for the support of these organizations and agencies which made my research possible, especially Isaac, muchísimas gracias por todos, and all of the mannequin genomics folk for broadening my research horizons, especially those that helped with the genomic and genetic efforts and collaborated with me on the sexual size dimorphism. And I wouldn't have a dissertation to defend without the hours and hours and hours of hard work of these amazing scientists who were willing to go out in the rainforest and have a great time, and then take all of the rainforest stained data sheets and turn them into data that I could analyze to answer some of these questions. It's been such a joy to work with all of these people, and I'm so fortunate to have gotten to do that. And then of course I have to thank Alice for this absolutely unbelievable opportunity. This has been a spectacular PhD experience, and I so appreciate your dedication and the time and effort that you have put into sharing your wisdom and helping me grow as a researcher. And of course I have to thank these adorable little fluff buggets who are so tolerant and just too adorable. And I wouldn't be giving this presentation without my family, who have just always believed in me and given me the confidence to embark on new adventures. So thank you so much. And then finally, I have to thank my fiance, Dave, who has been unfailingly supportive since the first day we met. So I couldn't have done it without you. And thank all of you for listening and letting me talk about birds. And I'll leave you with a picture of Costa Rica, and I'm happy to try to answer any questions you might have. Thank you.